everybody's been hearing about COVID antibodies, and I know Katie, you've been doing some research into COVID in general. I don't remember some of the specifics, but um, how would the vaccine create those that antibody response that we need,
Mhm.
and what is your research kind of touching on, and how is that all flowing together?
Yeah, so I think just to take a little bit of a step back, so just vaccines in general, kind of the goal is to do the training of the immune system, right? To get these cells that can recognize it and persist and produce antibodies without actually having to go through the infection itself, right? So to prevent you from getting sick, but still get all the training that the immune system needs. So the way this is normally done is you take the bits of the virus that you think are going to generate an immune response, and so it's not the virus itself, right? So you don't get the full infection, but you get the bits that the immune system needs to train it. So that's kind of the goal of most viruses. And so, the reason they take a long time to develop and, is that you need to know which bits are the important part. And then for things like the flu, there's this battle, right? So the flu is constantly changing every year. And so the vaccine that worked last year, now the flu virus has mutated that part, right? So now those cells can no longer recognize it and the vaccine won't work.
Mm-hmm.
So the part of, so that's, in general. And so there's a couple of vaccines that are being developed for COVID. And so hopefully we'll get one that produces a good response. Some things that I've been doing a little bit of is kind of tracking the immune response that's happening in patients without a vaccine. So patients that have gone through SARS-CoV-2 infection and have generated antibodies, what do those antibodies look like? And what's the T cell memory also look like? So kind of, have they generated kind of both parts of this? Do you need both the T and the B cell side? Is one going to be more effective than the other? And specifically, what are these name tags that we talked about of these cells that came from the infection? What do they look like? What do the antibody sequences look like that are capable of neutralizing the virus? And that will be important, for telling if vaccines are working. Can we generate those same types of responses in people after they receive the vaccine?
So what have you found so far, if a lot or anything really?
It's kind of in the early days, so I'll maybe keep you posted. But
Sounds good.
yeah.
So you mentioned the vaccine is not the full virus, right? You need the
Mhm.
important parts. So how would you manipulate, or how do the pharmaceutical companies or whoever is making the vaccine, how do they take the virus itself and get it down to those little important bits, so it doesn't actually infect you, and so your immune system gets the training, I guess, that it
Mhm.
actually needs? How does that work?
I'll let one of you two take that. Actually, don't know a ton about that part.
So there's just a whole host of ways is the
Yeah.
short answer. The reason that vaccines traditionally, I'll take a small tangent, but traditionally take years to develop is because it can be a bit costly. So they tend to run through one approach. Does it work? Doesn't it work? If no, try a new approach,
Mhm.
Mm-hmm.
right? And you just rinse and repeat until you find one of the approaches that does what you want it to do. The reason the
Okay.
vaccine development has been a lot quicker on this one is because a bunch of different companies are trying a bunch of different ideas in parallel, right? So the reason to do it
Okay.
in series is because if something fails, you can try one more idea, and that'll kind of keep your minimum trials, right? Maybe the third idea was the right one. I only tried 2 failures before. Alternatively, I can try 10 ideas right now, invest fully in all 10, and 9 of them might bomb, or maybe all 10 bomb, right?
Mm-hmm.
So right now we're having much faster development because governments and organizations are under high pressure to develop a successful vaccine now, independent of cost and energy, right?
Mhm.
So
Mm-hmm.
traditionally, the cost and energy part is sort of what slows down the R&D, as well as just basic research of, as Katie said, finding the particular molecule that matters. So maybe broadly speaking, antibodies react to proteins. That's kind of a key piece here. So the
Okay.
end of the day, you want to generate an immune response to a key protein on whatever your target pathogen is, if that makes sense.
Yeah.
So maybe we talk specifically about one of the clinical trials going on right now for one of the COVID-19 vaccines. I want to say it's
Yeah. Sure.
mRNA 1273 or something like that.
Yeah,
It's in a
Moderna.
phase.
Very, very specific.
Yeah, yeah. I was looking this up the other day. I
Yeah.
could be one of those numbers might be wrong, but it's pretty close. It's made by a company called Moderna, and they're in a phase 3 clinical trial right now. So this particular vaccine is called an mRNA vaccine, which is you heard in the name.
Mm-hmm.
So to maybe take a half step back, to understand an mRNA vaccine, we should probably just run through DNA, mRNA, protein for a second.
Go for it.
So DNA is in every cell, and it makes up the specific coding of you, right, in a nutshell. So think of it as, if you're on a construction site, this is the master blueprint. This is it. This is everything you can possibly do in a nutshell. RNA is when a specific page out of that blueprint is being copied a whole bunch and distributed to the different teams to do something, right? They're then going to read that copied page and they're going to put together a product, right? So to go back, it's DNA then becomes RNA, which is the specific copied piece, and then RNA gets translated into protein, which is the final product.
Okay.
Right. So this is how the traditional, what we call the golden rule, DNA to RNA, RNA to protein. And protein is what you're trying to elicit immune reaction typically to. So in this case, what this company has done is they've looked at the genome of the COVID-19 virus. Through other work and other assays, people have determined that it's what's called the spike protein is the most important protein we can find right now. And that this protein is what facilitates the virus getting into your cells where it can replicate and take over and cause the disease symptoms. So what they've done is they've found the particular mRNA code that codes for that spike protein. They wrapped it up in essentially a lipid carrier, and they're injecting it into people. And that the reason they're doing this is that, as we talked about earlier, you have macrophages, you have dendritic cells, you have B cells, and these fall under a class of cells called antigen-presenting cells. Which are very literal in their name. They will take antigen and they will present it to the adaptive immune system, which will activate to it and form a response to it. So the hope is that we inject these mRNA containing capsules into you. The antigen presenting cells will take it up. It is an mRNA, so they will then translate whatever it codes for, which in this case is the SARS COVID-19 spike protein. They will make this protein in isolation. It is just this protein. It's not the virus. It is a single part of the virus.
Right.
And then we'll present that piece of the virus to the T cells, who will then activate and form this immune reaction. They will activate B cells. You will get your antibody production, and you will form memory cells. Then later in life, if you go out into the wild and the actual virus gets into you, you have antibodies specific already for this spike protein. So the idea is that in this vaccine, we're actually not injecting you with a protein. We're injecting you with an mRNA transcript, which once it's in an antigen-presenting cell, will be endogenously, but the host cell itself will use its own machinery to build this protein, and then we'll take that protein and display it to T cells in a way that says, hey, you should react to this. That's the idea.
That makes
at least
sense.
behind this particular vaccine.
So we've talked about viruses mutating and coming up with different ways to infect you. Is coming up with a new protein a relatively common way for a virus to mutate, or is this going to be a really short-term type vaccine, or what are we kind of looking at with this?
Great question. So I guess the short answer is we'll tell you in 5 years.
Okay, fair enough.
The longer answer is, what you should know about DNA is that it's extraordinarily stable, i.e. if a mutation gets introduced, it usually gets removed and fixed. Not always, but usually. RNA is much less stable. And what I mean by that is if it gains a mutation, odds are it's not going to correct that mutation. That just becomes part of that virus going forward. And if you think about it in kind of a one of three possible outcomes, the virus picks up a mutation, and either one, it's helpful in that the virus is now a better virus. Two, it doesn't make any difference in the world. The virus is exactly the same. It has a mutation, but it has no functional difference. Or three, it is detrimental, right? If it's detrimental, that virus will not proliferate. It won't be as good. It'll get outcompeted by its counterparts. If it's better, the virus will now be better, and you'll see that strain start to take off. So we talked about the incredible specificity of your adaptive immune system, right? They can recognize down to a particular mutation within a protein. It's beautiful. The plus side to that is that you get a lot of highly specific responses. So like Chris was saying earlier, you can go up to a cell, kill it, and the neighboring cell is fine. The detriment to this is that you being the virus doesn't actually have to completely change a protein. A single small mutation that causes a little bit of difference can cause it to escape that highly specific immune reaction, right? So imagine that you are a CD8 T cell. You're walking around and we've taught you that you're looking for a red hat, a guy wearing a red hat. You're walking around, you find a guy with a red hat, you tackle him. Great. What happens when you come across a guy who's got a red hat that's been photo bleached and is a little lighter shade of red? Well, maybe you don't tackle him. That's not really the right
Oh,
color, right?
yeah.
So that one gets to keep walking.
Mhm.
He didn't have to take his hat off. He didn't have to change anything. It's just a little different color. So
Yeah.
viruses can do that, where they can actually just have a mutation that modifies a protein just slightly, and your adaptive immunity is gone. And this is what happens with the flu every year. So we make a
Mhm.
vaccine, you get antibodies to a particular protein, and then next year it's a slightly different version of the same protein, but your adaptive immune response can no longer recognize it. And now we got to start over with a new vaccine.
Mhm.
That makes sense.
So sometimes they ditch the protein entirely. Sometimes they just change it a little bit.
Wow.
You know,
Sounds like
there's a
the
penalty to being
vaccine
so
people.
specific.
Yeah, sounds like the vaccine people have a hard job for the flu. Come out with a new one every year. Huh.
Well, and
Yeah.
there's a funny thing to that story too, which I think a lot of people miss in sort of the pharmaceutical side of things.
Okay.
So the flu is a good example. You have to decide which flu to make in
Mhm.
like February, right, for next year, because you have to make it, right, and that takes
Right.
a while to make hundreds of millions of doses. So you have to predict ahead of time which drug you need, because you can't make it that quickly. And so there's a lot of that kind of going on in industry of going, well, I mean, would I like more time to test something? Sure, but the deadline's tomorrow, so pick one. And then off to the races you go, right? And that's why sometimes the flu vaccine is more effective than others, is that
Mhm.
you pick a direction, and then the flu, takes a hard jank left, and that's it, right? And then you missed it. So there's a logistical side to it that
Mhm.
tacks onto the science side of, well, how do I make a couple 100 million doses and distribute them
Mhm.
worldwide, and how long does that take? And then all the regulatory stuff that goes with that, right? You have to check it. There's a whole bunch of regulatory compliance stuff that comes with putting anything into another person.
Right.
Oh, yes.
So this,
Surprise, surprise.
yeah. So this just piqued out of my own curiosity, and if you don't know, that's totally fine. How do they pick which direction they're going to go when they're predicting the next one?
Yeah, so they're doing a lot of epidemiological data, right? So they're tracking a couple different versions and watching how they're developing in other countries
Okay.
that are in a different seasonal state than, for example, the United States and some of the more like the first world countries that are coming around to it. They'll have right, because we're on a different seasonal cycle than other
Right.
people, and so you can kind of like flu season is at a different place in different places around the world.
Mhm.
And so they'll watch, okay, what's going on here? What's the infectivity of this strain versus that strain?
Mhm.
What places is it starting to become a hot spot? And then there might not be a dartboard involved. That's really just throw something and see which one sticks.
Fair
But
enough.